So in the last class, we discussed purine theory, is not it? So also I gave you one assignment. To find out the frequency of heterozygous and homozygous individuals. To find out the frequency of individuals or plants having homozygous and heterozygous genes, isn't it? Do you remember? I gave you one to expand the binomial 1 plus 2 to the power m minus 1 whole to the power n. I gave you to expand this binomial with m is equal to 2, m is equal to 3 and n is equal to 4. After 3 generations of self-pollination, and when 4 genes are segregating, what will be the frequency of individual of plants having heterozygous genes and homozygous, different numbers of homozygous and heterozygous genes. So for here 2 to the power m minus 1 will be, if m is equal to 3, 2 to the power m means 2 to the power 3 is equal to 8. So minus 1 is 7. So this term will be 7, isn't it? This term will be 7. Then n is equal to 4. So to the power 4. Can you expand this binomial? Can you expand this binomial? The first term in the binomial, it denotes the number of heterozygous loci, heterozygous genes. And the second term in the binomial signifies the number of homozygous genes. So when you expand this binomial, anybody can do it? Anybody can do it? So have you got the small binomial like a plus b whole square is equal to how much? A square plus twice ab plus b square. Then a plus b whole cube, a cube plus b plus. So now you have to expand a plus b whole to the power 4. Can you do it? What will be the first term? What will be the first term? This will be the first term in the binomial expansion, and the second term will be 4c1. Good. Third term will be fourth term. You know it. You know it. You know it. Now you solve one by one. And here you see in the first term, that is to the power 4. This indicates that there are 4 heterozygous genes. So that the solving entire term will give you the number of individuals having 4 heterozygous genes. Okay? As I told you, the first term indicates in the binomial expansion 1 and 7. First term is 1, second term is 7. The first term gives the number of heterozygous genes. Second term gives the number of homozygous genes. So here in this term, 1 to the power 4 means 4 is the number of heterozygous genes. So 0 is the number of homozygous genes. That means, and you solve this term, this will give you the number of individuals or plants having 4 heterozygous genes. There are 4 genes, is not it? 4 genes are segregating. That means originally two parents had gene differences for 4. So this term will give you all if you solve this term, this will give you the number of individuals having 4 heterozygous genes. And if you solve this term, this will give you the number of individuals having 3 heterozygous genes and 1 homozygous genes. And if you solve this term, third term, it will give you number of individuals having 2 heterozygous genes, 2 homozygous genes. Likewise, this will give you the number of individuals having 1 heterozygous gene and 3 homozygous genes. And if you solve this term, it will give you the number of individuals having all the genes in homozygous conditions. All the genes in homozygous conditions. I gave you that to find out the number of homozygous individuals, the proportion of or frequency of completely homozygous individuals, where after m generation of selfing, when n genes are segregating, was given by how much? Is given by 2 to the power m minus 1 divided by 2 to the power m, the whole to the power n. So this term, the last term. You solve which term? Can you solve the first term? What will be? Factorial 4 divided by factorial 0, factorial 4 minus 0, factorial 4. So factorial 4 cancel, factorial 0 means 1. So ultimately this term will be happy. If you solve it, this term will be 1. Good. There will be 1 individuals with all the 4 genes in heterozygous condition. Okay? Then the second term, if you solve the second term, it will be factorial 4 divided by factorial 1 into factorial 4 minus 1, factorial 3. If you solve this, this will be 4 into 7, 28, isn't it? So it will be 28. If you solve the third term, it will be 4 factorial 4 divided by factorial 2, factorial 2. Isn't it? How much it will be? And 49, this will be 7 to the power 2, is not it? 49 multiplied by 6, how much it will be? Multiply it. 294. Good. 294. Then the fourth term, factorial 4, factorial 3, factorial 1, isn't it? Am I correct? And it will be 7 into 7 into 7 multiplied by 4. How much it will be? 1372. Good. This will be 1372. And the last term, factorial 4, factorial 0 into so this cancels 7 to the power 4. What will be 7 to the power 4? 2 4. Good, very good. 2401. Now you have the data, required data. Individuals having 4 heterozygous genes, 1. Number of plants having 4 heterozygous genes, 1. Then 3 heterozygous genes, 28. 3 heterozygous genes and 1 homozygous gene. Okay. Then 2 heterozygous gene, it is 294, isn't it? One heterozygous genes, it is how much? It is 13. 0 heterozygous gene. That means all are 2401. 2401. That is completely homozygous individuals. Frequency of completely homozygous individuals. So this you can also work out using the formula that I have already given at the top. 2 to the power m minus 1 divided by 2 to the power m whole to the power n. You put m is equal to 3 and n is equal to 4. You will get this. Okay? This is what happened when we hybridize between two diverse parents having differences in gene numbers. So you see, this is the minimum population. The total of this, if you total up this is the minimum population. What will be the total? You total it. Total it. 1 plus 28 plus 294 plus 1372 plus 2401. You total it. How much it is? How much it is? Total? 4096. 4096. After 3 generations of self-pollination, when the two parents differ for 4 genes, then you need to go, the minimum perfect population is 4096. If there is only 1 gene, and if you go for 1 generation of selfing, it was 4, is not it? It was 4. So you see, this is the minimum perfect population, but in the practical field, when you go to determine, to see the inheritance of a particular character, you have to go much larger population. If 2 genes are segregating, minimum perfect population is 16, 4 to the power n, is not it? 4 to the power n. 4 into 4 is equal to 16. 1 gene means 4. 3 genes means 4, 4, 4. 64, is not it? So this is the minimum perfect population, but in reality, when you go for studying the inheritance of a particular character, then you have to go much larger population. Much larger population in order to score the different phenotypes, and then that will be subjected to chi-square test, whether a particular ratio is fitted into the observed data or not. And it is the task of the breeder to select the best recombinant, because he is always searching for the best recombinant, isn't it? The genes of the two parents must combine to give a recombinant which will be having all the desirable genes. Into one genotype. So breeder has to select that one. So breeder's task is very, very tough. because he has to do everything on the basis of the phenotype. So now let us start from the last class. We discussed about the PO line, is not it? PO line theory. What is a PO line? What is a PO line? What is a PO line? Who can tell me? What is a PO line? It is the progeny of what is a PO line? What is a PO line? I repeated what is a PO line, this definition, I think 20 times. What is a PO line? Who can tell it nicely? What is a PO line? It is the progeny of a single homozygous plant in a self-pollinated species. So, any variation within a PO line is purely environmental. So, selection within a PO line is not effective. Johansen did his experiment using a commercial seed lot, and he found that the commercial seed lot has genetic variation. That is why he was successful in isolating 19 different PO lines having different seed sizes. And he also confirmed that the variation within the PO line was entirely due to the environment. It has no genetic basis, but the original seed lot has genetic variation. That is why selection was effective. Okay, but a PO line may get impure. A PO line may get impure with time. If you cultivate a PO line for a long time, there may be variation within the PO line. Then what are the sources of variation within a PO line? Sources of variation in a PO line. Mutations. These are the sources of variation in a PO line, because during the course of cultivation of the PO line, it may get mixed with other PO line seeds during sowing, transplanting, harvesting, threshing, storing, transportation, all these operations, it may get mixed with other PO line. And when it mixed with other PO lines, that will lead to variation. Okay, that is one source. Then a second source of variation is the natural hybridization. Already you have come to know that even in self-pollinated crop, there is some outcrossing or cross-pollination. For example, in rice there is less than 5% cross-pollination. In green gum there is 0.5 to 3% cross-pollination. Likewise, in other crops also there is some amount of cross-pollination, and it depends upon the environment in which it is grown. So, depending upon the environment, there may be some amount of cross-pollination in the self-pollinated crop species also. So, this outcrossing or cross-pollination in the self-pollinated species may lead to genetic variation. When there is natural hybridization with other varieties, then definitely there will be some genes transferred into the pure line. So this will lead to variation. Then the third factor is chromosomal aberration. In self-oriented crops like wheat, barley, oats, there is found some chromosomal aberrations. There is formation of, there may be duplications, there may be deficiencies. There may be, so as a result, there may be meiotic abnormalities, formation of breach, formation of laggards, fragments. So these chromosomal aberrations, if there are small cryptic, small differences in the structure of the chromosomes, then these are often not detected. But in sometimes they may be important. For example, in wheat, oats and barley, these type of chromosomal aberrations have been detected, and they are also playing a role in creating variation in the pure line. Then last but not the least, this is the most important factor, mutation. Mutation is the most important factor in the evolution. Mutations. So what is mutation? Can anybody tell? So it is the simple definition is it is the sudden heritable change in the character of an individual or plant. Okay, and when there is a chemical change in a gene, it is referred to as point mutation or gene mutation. Point mutation or gene mutation. It refers to a chemical change in a gene. Chemical change in a gene. So this point mutation or gene mutation, actually it is the major sources of variation in the crop plants. Almost the entire variation found in the crop plants have the origin in mutations. Sometime during the course of evolution. These mutations occurred have led to the variation in the crop plants. But these mutations, this become important with time. If you grow a crop variety of pure line for long period of time, there is the mutation may get importance because the frequency of mutation is very, very low. The mutation frequency estimated in maize. So Stadler 1942 estimated the spontaneous mutation frequencies in maize, and he found that it is of the order of 10 to the power minus 4 to 10 to the power minus 6. 10 to the power minus 4 to 10 to the power, actually it should be like 10 to the power minus 6 to 10 to the power minus 4, is not it? 10 to the power minus 6 to 10 to the power minus 4. 10 to the power minus 6 to 10 to the power minus 4. So minus 4 is greater than 10 to the power minus 6, is not it? Minus 4 is greater than 10 to the power minus 4. So range is 10 to the power minus 6 to 10 to the power minus 4. This Stadler, he estimated the spontaneous mutation frequencies in maize. Similarly, in other crops also, the spontaneous mutation frequency is very, very low. It is generally of the order of 10 to the power minus 5 to 10 to the power minus 6 to 10 to the power minus 6 to 10 to the power minus 5. It is very low. That is one. And another mutation is generally recurrent. The one character, it is suppose get mutated, so again reverse back sometime in the course of the evolution, future course of evolution, it may get revert to the original character. Okay, so that is why the mutations in the short period of time, it may not be important, but in the long run, so mutation may be important. Mutation may be important in the long run. Consequences of self-pollination. Actually, we have discussed already this one. When we discussed the consequences of genetic consequences of hybridization. When you self, then what happens? So self-pollination has two effects on the population. Number one, there will be a rapid increase in homozygosity. There will be a rapid increase in homozygosity. That I think you have already come to know from the discussion on genetic consequences of hybridization. If you consider a single locus, so this is a heterozygous individual, consider a single locus. If you self-pollinate it, self-pollination, then what will happen? There will be 1/4 AA, half capital A small a is to 1/4 small a small a. Then what will be the proportion of homozygote here? Half 1/4 plus 1/4 is equal to half. See, if you go for next generation, if you go for another generation of self-pollination, this is after one generation, second generation, first generation, this is second generation. What will happen? Go through it. What will happen if you go for second generation of selfing? The population, the entire population, these plants will be self for another generation. Then this is homozygous, is not it? So this will breed true. Because it is homozygous, so this will breed true. This proportion will breed true. This will remain the same. Other factors remaining constant, like the survival of each and every individual is assured in the population. If survival of each and every individual is assured, if gametic proportions are equal for all the alleles, okay, if there is no competition under these conditions, this will breed true. This will remain the same, 1/4 capital A, capital A. Similarly, this proportion will also remain the same. This will breed true, 1/4 small a small a, 1/4 small a small a. Then this will only segregate. This will be again 1/4 capital A, half capital A small a, and 1/4 small a small a. So this 1/4 actually this is from half. So you have to multiply it by, these are proportions, so you have to multiply it by your half. So half into 1/4 will be 1/8. Half into half will be 1/4, and 1/4 into half will be 1/8. Then you find out the proportion? 3/4. You see, 3/4. 3/4 means 75%, and half means 50%. So there is rapid increase in homozygosity. Is it clear? The first statement, that self-pollination leads to rapid increase in homozygosity. We have seen that after first generation of self-pollination of an individual heterozygous for a single gene, it is half that is 50% reduction in heterozygosity, or in turn 50% increase in homozygosity. In the second generation, it is 75% homozygosity. So there is rapid increase in homozygosity. Have you heard from the back? Is it visible from the back? Last one. What's your name? Is it visible? Then the second effect of self-pollination on the population is differentiation of the population into distinct homozygous lines. Differentiation of the population into distinct homozygous lines. That has already been discussed when there is, if we consider a single locus, capital A, after first generation of selfing, there will be capital A, and small a. Okay. This is 50%, then gradually this heterozygosity will be reduced, and after 5-6 generation, more than 90% of the individuals will be homozygote. So the population will have mostly two types of individual, two distinct homozygote. One will be capital A, the others will be small. Likewise, if you consider 4 genes, there will be how many homozygous possible? How many homozygotes? If there are 4 genes, you work out how many homozygous possible. If there are 2 genes, how many homozygous possible? After 5-6 generations of several generations of self-pollination, how many homozygous possible? Can you work it out? One is this. The other one is this. Then 4. Correct? So you can also work it out like this. So this will be small a, capital A. If there is a third gene, how many? Homozygous? 8. 8. Then what is the generalized formula? If there is 1 gene, if there are 2 genes, if there are 3 genes, that means 2 to the power. I gave you this table. Why don't you remember? Who gave the pure line theory? Huh? Loudly. Speak loudly. Huh? Johansen. In the year 1903. Johansen gave the pure line theory in 1903. Johansen used the commercial seed lot of the crop. Not all, I will ask. Johansen used the commercial seed lot of the crop. The last one. France bean. Its botanical name? Phaseolus vulgaris. Huh? So what was the variety? Variety? No, not all. I will ask. What was the variety? What was the variety? Princess. Princess. The princess variety of Phaseolus vulgaris. What is the mode of pollination of Phaseolus vulgaris? It is strictly self-pollinated. Strictly self-pollinated. So you have come to know what is pure line, what is genotype, what is phenotype. From this Johansen classical experiment, we came to know that genotype, phenotype is the result of genotype plus environment. Okay? Genotype plus environment. So in terms of variance, this Vp is equal to Vg plus Ve. So we have also come to know that the portion of the genetic variation which is inherited from generation to generation, one generation to the other and next generation, the portion of variation that is inherited from one generation to the other is known as the heritability. The proportion of genetic variation that will be inherited from generation to generation is the heritability. So it is the proportion of genetic variation to the total variation. Last class I gave you. So it is heritability in broad sense. Vp divided by, Vg divided by Vp. It is expressed in percentage 100. Okay? Heritability in broad sense. So for selection to be effective, there must be variation, and a variation must be heritable. There must be variation, and the variation must be heritable. Only then selection will be effective. So we may be interested to know what will be the gain, or advance when we select in a population of pure lines, a mixture of pure lines as used by Johansen. Johansen was successful in selecting or isolating 19 different pure lines with characteristic mean seed weight ranging from 640 milligram in line number 1 to 350 milligram in line number 19. He was successful because the variation was genetic. So variation must be there, it must be genetic variation. So we may be interested to know that what will be the advance or gain if we select in a population which is a mixture of homozygotes or which is a mixture of pure lines. So that the improvement, in other words, the improvement improvement in mean genotypic values of selected families over that of the original population or the base population over that of the original population is known as the genetic advance under selection. Genetic advance under selection. Genetic advance under selection. So as used by Wanchen, the breeders of the different crops, they also use a population in self-pollinated crop. They also use a population which is having variability, and generally these populations are the land varieties, land races or local varieties. Is it complete? Population of self-pollinated crop land race or land varieties. Land races or land varieties of self-pollinated crops. Actually, these are the varieties which have been under cultivation since long, and as a consequence of their long history of cultivation, the forces that we have already discussed, mechanical mixtures, natural hybridization, chromosomal aberrations and mutations, are acting on the population, and it leads to variation. Because there is self-pollination, so that means the population is a mixture of homozygotes or pure lines. So breeders, when he opted for selection in a self-pollinated crop, he first used a land variety or land race. So the land race is nothing but a mixture of pure lines. Mixture of pure lines. So we can calculate, because this is the mixture of pure lines, we can select the different plants and grow them in progenies, individual progeny rows. We can select individual plants for some character in which the breeder is interested in. Maybe earliness, maybe stature of the plant, maybe seed size, maybe panicle, length, grain size or seed size, different characters, maybe tiller number, number of branches, leaf color. There may be lot of characters are there in which breeder is interested. He will go for selection in this population. If this population is having the variation, so he will select individual plant and grow them in progeny rows, individual plant progeny rows. Okay. Then he will look for variation within the progenies. He will look for variation within the progenies for the character for which he is selecting. For the characters for which he is interested in. So for the character in question, he will look for variation. If there is no variation, it will indicate that the original selected plant was homozygous for that particular character. Okay? If there is no variation, all individuals are similar, then this will indicate that original plant was homozygous for that particular character. If there is variation, he will again select within the progeny if founds some outstanding plants for the character in question. Okay? So in this way, he can select a number of plants and he will multiply the seeds as Johansen did for his pure experiment. Then he will go for yield trials. But he may be interested at the initials that what will be the advance expected when we select for a particular character in this population. And for that, he can estimate by growing it in a replicated trial. I will explain it later on. He can estimate the VP and also VG and VE. This he can estimate. And from this, he can also estimate the heritability in broad sense. That is VG by VP. Okay? So he will be having this phenotypic variance. And using these parameters, the breeders can well ahead of time predict how much gain or advance that can be made by selecting in that population for a particular character. So that already we have given that genetic advance under selection, and that is given by K into sigma square P into heritability in broad sense. So this sigma square P I have written, this is nothing but the square root of the phenotypic variance. So you can also use VP. You can also use VP. It is the square root of the phenotypic variance, that is total variance. At this stage we have the total variance is composed into two. It is it is composed of two terms. One is the genetic variance, other one is the environmental variance. Okay. So when we take the root of the phenotypic variance, that will be, you have to give the root sign. Please give the root sign. Square root VP. Square root VP. Okay. Square root VP. So this again you can simplify it, K into so this again you can isn't it? Is it okay? We have just the heritability in broad sense we have put in terms of VG by VP. So VP, square root of VP, square root of VP will be VP, isn't it? So this is square root of VP. This cancels. So we have VG and square root of VP. So this K is the selection differential. Selection differential. Axis, which is nothing but the standardized selection intensity. Standardized selection intensity is the selection differential. So at different selection intensity, we can work out the selection differential. So for 1% selection intensity, the K value is 2.64, and for 5% the K value is 2.8. 0.04. So this you can have from table, this value, K value, that is if you select 1%, what will be the expected advance? If you select 5%, what will be the expected advance? You can also go for 10%, the value is there. So simply by putting these values, say for example, if you 5% selection, if you select 5%, 5 out of 100 plants, out of 100 plants, if you select 5 in that population, what will be the genetic advance? Based on the estimates we have made already, heritability in broad sense and phenotypic variance. So it will be putting this figure, you calculate let us take it 9, so that you can take the root, and you take it 6 and 3. Okay, 9 is equal to 6 plus 3. Vp is equal to 9, 6 plus 3. We calculate Vg will be 6, is not it? This is 3. .12. 4.12. So unit will be same. If you are using it in centimeter or maybe number, maybe gram, kg, whatever the unit, the same unit will be here. Okay. And you can express this, how much will be the gain in terms of the original population mean. Suppose the original population, the base population, original population has a mean of say 20 grams, say for example, gram per plant yield, mean, grand mean of that population. You can express it in terms of the original population mean. That is 4.12 divided by 20 into 100. Calculate it, how much it is. Calculate it, how much it is. Hmm? 20.6. This means if you select 5% of the individual, 5% of the top 5% of the individuals for a particular character, say for yield per plant, then you can expect 20 point, how much it is? 20.6% gain over the original population. This is expected. 20.6% gain over the original population. When you select based on yield, 5% plant, you grow them and you find out the yield, actual yield performance. It may not be exactly 20.6, but it may be 16, it may be 22, something else. And that variation happens why? Because genotype is also affected by the environment in which a particular genotype is grown. That means environment also influences the expression of the character. That's why that fluctuation may occur. You may not get exactly 20.6, you may get less than this or maybe higher than this. If it much fluctuate, maybe suppose you are getting 20.6, it is about 40. In some cases it is 10 or less than that. You will understand that this character is much more influenced by environment. In that case, the population is, you are advised to grow the population in several locations to find out the genotype environment interaction. We will talk about this in a later class. Okay, if you have any question? Any question? If you have any question, because this will be continued in the next class, so you can also ask me in the next class. Okay? Next class we will talk about in details. Also we will discuss, repeat this last portion, genetic advance under selection. Okay? Do you have any question? No? No? Next class you will ask. Huh? Huh? X bar is the mean of the original population, that is. The population that which you are using for selection. That is the original population or base population. And that population will have a mean, is not it? For that particular character. Suppose grain yield per plant. So you have to take sufficiently large number of plants and you measure the grain yield per plant. You take the average. That will be the mean of the population. Okay, if suppose there are 1000 plants, you can draw 10 plant sample from different parts of that experimental area and you find out the mean. That will be the population mean. Out of 1000 plants, if you take 10 plants, sample plants and take the weight, that will not represent the population. That is the sample mean out of a population. But to represent the population, you will have to take sufficiently large population, representing the entire population, entire experimental area. So as to take care of as well as the variation in the microenvironment. Okay? We will again discuss these things. If you have any question in specific question in mind, you can ask immediately, bullet type question, and also we can discuss in the next class because the time is over. Okay? Okay? Thank you.